Hello everyone, welcome back to our weekly video from This Is Not a Science channel, and today we will be talking about enzymes. Since this is our biology video, enzymes one of the most important molecule in our body that just keeps life going, and it's amazing to learn about it. So let's start. Before we actually jump into understanding what enzyme is, we have to first realize the concept of what a catalyst is. So what is a catalyst? Here's a definition of a catalyst. It is a substance that increases the rate of a chemical reaction and is not changed by the reaction, which basically means it gives a boost to the reaction, but it's not actually involved in it. And this is good because we actually don't want the product to be affected by a catalyst. So it is just there, it's helping the reaction to go faster, but it's not involved in the product. So now we can finally answer the question on what is an enzyme. Well, here's the definition. It is a protein that acts as a biological catalyst, and it is really important to note that all enzymes are made out of protein. And the reason why we call it a biological catalyst is because it catalyzes biological reactions, which is all around our body. That's why we have loads of different types of enzymes in our body to speed up the reaction so life could be possible. This brings us to the question of why are enzymes so important? Again, there are loads of reactions going on in our body and we need enzymes to speed those up, right? If those reactions are not speeded up, it will be way too slow for life to be possible. For example, if you're breathing something, there are at least 4 or 5 types of enzymes responsible for your breathing right now. For your gas exchange, if those enzymes do not exist, there is no way for you to breathe properly in order to support your life. Same thing goes for digestion. All the other systems in our body require enzymes to speed up the reaction. Now we can look into the structure of an actual enzyme. So here's the enzyme, and there's one special spot that we have to mark, and it is this corner right here. We call this the active site. And the active site is where the enzyme receives what it has to process. And we call this active site as being complementary to its substance. So it is very specific. There are different types of enzymes with different active site shapes, and they can only fit in a specific type of molecule. It will not fit in other type. The reaction will not work if the substrate does not fit the active site. Now we can actually see an enzyme in action. So before that, we have to understand the concept of substrates and enzymes. Here we have our enzymes. Alright, these are our enzymes. And we have to make sure that the reaction starts by inserting something called a substrate. The substrate is basically whatever goes in the active site of the enzyme in order for it to process. So we can either have a substrate where we have to break it down, or substrates where we have to join them together. So an enzyme can break down or join substances. In this case, we have the red substrate right here. The enzyme is responsible for breaking it, and we have the blue substrates right here. Enzymes is going to join it. Then we have to look at something called enzyme-substrate-complex and their products. So once the substrates have successfully fitted in the active site, when they are actually combined in one piece, we will call this an enzyme-substrate complex. And it has to be this specific name because it's enzymes and their substrate successfully joined together. So after they have joined together, we will get our product. In this case, we are breaking down the red and we are joining the blue. So this is what we call the product. And here are some examples of enzymes for you guys to picture this easily, what enzymes actually do in our body. So let's take digestion for example. We intake a lot of different biological molecules in our meal, and we have mentioned that in the previous video, so make sure you check out that previous video about our biological molecules in order to figure out what enzymes actually process. So we have enzymes such as amylase. Amylase is a type of enzyme that converts starch into monosaccharides, which is right here. Starch, if you remember from the previous video, is that complex ring structure tightly packed together, and we'll separate them into single sugar molecules, which we call monosaccharides. And amylase is the exact enzyme that breaks it down. And we also have protease. Protease, they convert proteins into amino acids, and protein is a huge part in our diet. So anything such as meat and fish or egg, those contain proteins, and we need protease to break these down into single amino acids. And we also have lipase. Lipase is to convert lipids into glycerol and fatty acids. So again, from my previous video, this is how a fat molecule actually looks like. On the top we have a glycerol connected to 3 fatty acids. And without lipase, there is no way that we can break this down successfully and efficiently. So these are the main types of enzymes in our body responsible for digestion. And obviously there are a lot more enzymes catalyzing different reactions such as respiration or excretion. So those are the enzymes that will come later in more advanced content, but these are the 3 that you mainly have to remember for now. Now I can provide you with some additional information about enzymes. So you might ask, how do enzymes and their substrates bind? Well, they're not some artificial intelligence or anything, alright? They do not have the ability to find their substrate that fits. It is all random collisions, right? So for example, if we are digesting something that requires enzymes to break down, in for example protease have to randomly collide with the food molecules to find the correct substrate, which in this case will be protein. So everything is random, it depends on chances. And the enzyme will successfully catalyze a reaction only if the collision is successful. What does that mean? It means that the substrate has to perfectly fit in the active site. This is what we call a successful collision. In the other case, where the substrate did not really fit in the active site perfectly, we will call this an unsuccessful collision where no reaction will happen. So everything is depending on chances. In the next video, we will discuss about how temperature and pH, acidity or alkalinity, affect the rate of enzyme reactions. So that's it for this video about enzyme, and make sure you like my video and subscribe to my channel, share it to all your friends who hate science, and remember, these really motivates me to keep on making new videos weekly just to teach more people about these essential biological, chemistry and physics knowledge. So make sure you like the video and stick to me, and our next video on biology will be about how enzymes further proceed under different conditions. So thank you very much, see you next time.